Good afternoon, ladies and gentlemen, and welcome to this afternoon's session. When we were planning this session, we were talking about the 4 different dengue viruses, and those who know me know that I don't like calling them dengue 1, 2, 3, and 4. I like calling them Tom, Dick, Harry, and Frank. And that was what I wanted to actually call this session, an analysis of Tom, Dick, Harry, and Frank, but I got outvoted. So the title of this session is Are All Dengue Virus Strain Serotypes Created Equal? And our first speaker is going to be Eng Eong, and Eng Eong is going to talk about dengue virus genetics, not Loki, for Endgame. Over to you.
Thank you. Thanks, Alan. And thanks very much to Adam and Stephen for the opportunity to be here and share these thoughts with you. So very unlike Alan Rothman, who looked at the word Endgame and thought of chess, I thought of Marvel's Avengers, clearly. Right? But I thought what I'll try and do is go and try and directly answer the question that Alan Barrett had just posed. Are we dealing, this idea that we have dengue virus with 4 serotypes, or do we have 4 different viruses? And so that's going to be the topic of this talk. So the first half is based on a published paper. It came out last year, but I just want to provide the context for perhaps a debate that has gone on. And for those of you who are older will remember the debate that we had in the field. And then in the second half will be a set of very preliminary data. We just started working on this. But I thought that what we found was just too intriguing not to share. So what you will see in the second half is very preliminary. And I will take it from there. So as many of you might, or some of you might remember, we had this big debate between 2 schools of thought. One was obviously ADE as the primary mediator of, mediator of risk for severe disease. And then there's a second school of thought that said that it's actually the virus, it's not the strain. And essentially, Leon Rosen actually belonged to that second school of thought. And this is a very interesting paper. I would recommend it to you because it was, I think, written with punches pulled, and he concluded, simply stated, that different strains of dengue virus of all 4 serotypes vary in their pathogenic potential, that you don't need ADE to explain severe dengue. And essentially, the whole debate started because of this event. Prior to this, of course, the idea that ADE is an important pathogenic mechanism was proposed by Halstead and others. Then came this outbreak that started in French Polynesia in Orange. So in Orange is 1971, and in 1972, the islands that had these outbreaks were in color in yellow, and then finally in Tonga in green in 1973. And it started with a big outbreak in French Polynesia that then moved to Fiji, and that caused another explosive outbreak. And then the virus moved to islands of New Caledonia, Niue, and Samoa. And New Caledonia and Niue, there were still big outbreaks. And it was in Niue that Leon Rosen first described primary dengue 2 in children who had very severe hemorrhagic disease. Right? And that's why he said that you don't need secondary infection to cause severe dengue. But by the time the virus moved to Samoa, the disease became less severe. Duane Gubler, who was part of the team at that time, anticipated that the virus would get to Tonga, and he himself went to Tonga, set up a surveillance system, and every day according to him, he would go to the hospital and look for admissions that could be dengue, but he didn't find many. In fact, the viruses that he isolated from Tonga were from people with very mild fever cases. So it seemed like as the virus moved from island to island, it became more and more attenuated. And so that was the idea for a long time until years later, Shannon Bennett and with Argan Steele, who was a PhD student then, went back to the viruses that were isolated during that time and then did a sequence and found that in Tonga, viruses, they separated into a separate clade compared to those in the other Pacific islands, and shown in panel A on the left-hand side. So what you see in the bottom part of the tree are those viruses colored in green, and so those are the Tonga virus, and those in the yellow and orange came from those from other islands. I want to point out 2 things here. So in red arrows are those mutations that separated the Tonga virus from those from the other islands. But there was a blue arrow, and that's Tonga 74, which was then used as an attempt to make the dengue 2 vaccine by NIH. And Tonga 74 lacked PRM mutation. And I'll come back to that story in the later half. But essentially then the question is Tonga virus, can we see an attenuated phenotype in the lab? And so we use a plug assay. Now, it's not a great representation of what happens clinically, but at least the epidemiological phenotype had been described. So now we can use this in vitro phenotype to kind of try and get a handle on what the mechanism might be. So in the top 3 plug assays that you see, NC stands for New Caledonia, FJ is Fiji, and PF is French Polynesia. Those are the plugs that will form with these isolates, whereas in the bottom panel and in the boxed up in rectangle at the Tonga virus, and you can clearly see that the size of the plugs were considerably smaller. And on the right-hand side is the statistical analysis for that. You can see that those viruses starting with T have formed smaller plugs. So then the question is, which of those 3 mutations that separated Tonga from those on the other islands gave rise to this phenotype? So now we took the New Caledonia virus and we did single substitution of these 3 mutations into the New Caledonia backbone. And as you can see in the bottom, on the left-hand side, the bottom, the photographs, is a PRM histidine to arginine substitution at position 86 that reduced the plug size, right? And then on the right-hand side, you can do the opposite. You can take the Tonga virus and then reverse the mutation from histidine back to arginine. Arginine back to histidine, and you increase the plug size. So something is, this PRM mutation is somehow attenuating infection in vitro. And so I'm going to cut a very long story short because this is a published paper from last year. But essentially the mechanism is this, that the codon used to encode arginine, at least in the Tonga virus, compromised translation. Right? And so what you see on this Western blot is that the 3 from top to bottom is NS3, E, and PRM, and then the beta-actin is the control. You can see that New Caledonia, you're seeing all the proteins expressed, but in the Tonga virus, at this 24-hour time point, you see a very delayed translation of the proteome. If you take the New Caledonia virus, so the one in NC, and then you do the PRM substitution into the New Caledonia backbone, you reduce translation. If you take the Tonga virus and you reverse the arginine back to histidine, you increase translation. So the codon encoding this arginine residue is attenuating a mutation. So, and now exactly how that is probably due to tRNA modification, but we didn't go into that part of the mechanism. The question we asked instead is, how would a virus that is so weak be selected for and actually got into Tonga in 1973? The answer is because the same virus, the same substitution, made the virus very fit in the mosquito midgut, and only in the midgut. If you take this virus and inoculate it directly in the mosquitoes, you don't see any difference. And the reason why this substitution gave greater fitness to this greater fitness of the virus in the mosquito midgut is because the arginine residue is positively charged. And the mosquito midgut, unlike ours, is alkaline, not acidic. So in an alkaline pH about 8 to 9, then only arginine will remain positive, whereas histidine would have lost the positive charge. And so the positive charge of the arginine allows the virus now to attach onto the negatively charged receptors on the midgut to facilitate infection. So that part of the story was in the main part of the paper. What's in the supplement, what's in the supplement part of the paper, which I thought for today's discussion could be interesting, is this. If you take Tonga 74, which lacked that PRM mutation, and now you substitute the histidine to arginine in the Tonga 74, you do not make any change to the plug size, right? And that's because the Tonga 74 has 3 other mutations that are boxed up in red. And those 3 substitutions could potentially now alter your tRNA modifications and therefore change your codon usage in the infected cell, right? So even though they all belong to that same clade, the fact that you have one or two additional, or in this case, 3 additional amino acid substitution could completely change what the kind of host virus interactions that go on to determine whether it's going to be pathogenic or whether it's going to be attenuated. So, and this Tonga, the events in the South Pacific are not the only instance where a small number of genetic changes could give rise to very big phenotypic differences, at least in the epidemiological context. And so this is a review written by Alison Choi, who all this was done as a part of a PhD. There were several others. I think the one on the left is the 1997 outbreak in Santiago de Cuba. Actually, the clue for this mutation came from Scott Halstead himself, that said you should look at NS1. And we did, and we found that the substitution NS1 changed the lipid content of the hexamer that is that's secreted into the plasma. And so that potentially could explain the difference in the disease outcome, but it also increased fitness in the mosquito. And then on the right-hand side, as where I started because of this push from Duane Gubler to go and sort out the genetics of dengue virus, with the events of 1994 in Puerto Rico, where actually the dengue 2 was introduced in 1982, but the outbreak didn't happen until 1994, and it needed to acquire a series of mutations to give rise to a fitter virus, and the mechanism is in the paper, so I won't get into that. But I think the point I want to make is that we think of dengue as one virus, and then 4 serotypes, when in reality I think we're dealing with 4 different viruses, and within each of those types are very different lineages. So we shouldn't be thinking serotype or genotype. Maybe we should be thinking more in terms of the minor lineage, because that's where the interesting things will be, or if we want to understand epidemiology and clinical outcome. And so in the last 9 minutes or so, I'm going to now convince you from a very preliminary set of data that one of the reasons why it's so difficult for us to understand pathogenesis and immunity is because we are dealing with 4 different viruses, right? And I think Eva Harris just kind of said this many times in her talk and as well as in the Q&A. And so in the left-hand side is a tree of orthoflaviviruses, and the silhouettes are the hosts, the mammalian host or the avian host for these viruses. So if the box up in the red is dengue, and if I blow it up, then you see it on the right-hand side. The point I want to make is this. The branches that separate dengue 1, 2, 3, and 4 is about the same as those in blue. Yet in blue, we call them different species. In red, we call them serotypes. So I think genetically, they are more similar to species level differences than just one species with 4 antigenic types. And so to show you that now, at least in the context of host response, these are very different viruses. So this is the experiment, and this was done, I think, I believe, earlier this year by Kivan Kumar, who's here. So we took whatever viruses that we had, dengue 1, 2, 3, and 4 in the lab, and basically used this to infect A549 cells, each one in triplicate, and then we redid it because I couldn't believe the data. So we did it twice. So the isolates that we got were not all from Singapore. So these came from Indonesia. They were at different times, including the prototypic viruses that were used to develop the NIH TB003 vaccine. And some of these were rescued from infectious clone. So when you see dengue 1, 2, 3, and 4, they were not all from Singapore viruses. They were isolated in different geographic regions over different periods of time. And then on the right-hand side, this slide is just to orientate you for what's to come. So the mock infection is in leftmost, and then in light blue is dengue 1, yellow is dengue 2, dark blue is dengue 3, and then in orange is dengue 4. So everything that you see now from there on, it's always in that order, and the red dotted line is the mean of the mock control. Okay? And in the interest of time, I'll just focus on cellular response to type 2 infection and antigen presentation. So we know about T cells. This is CD8 T cells. Classically, it will be recognizing proteins that's translated in cytoplasm, which is then processed by the proteasome. And just to again orientate you to what's come, the proteasome can be divided into what is constitutively expressed and what is induced by interferon gamma. And so the interferon gamma-induced proteasome are called the immunoproteasome. And then the peptides are then transported across the endoplasmic reticulum by TAP. It's then trimmed by the protein ERAP, or endoplasmic reticulum aminopeptidase, and then docked onto MHC class 1 and then presented to CD8 T cells. So these are the HLA molecules, and you can see that for dengue 3, it's the lowest. Dengue 4, followed by 2, are higher. Right? 1 is a big spread, but dengue 3 is quite tight, despite the fact that these are constructed from a sequence from different time periods. It's not just HLA, but the proteasome, and these are the immunoproteasomes, are also lowest for dengue 3. And it's possibly low because interferon gamma receptors, and these are A549 cells, so they don't secrete interferon gamma, but they have the receptor, and it's also low for dengue 3 infection. And if you look at the TAP genes, they are also low. And finally, the aminopeptidase that needs to trim the peptides to be docked onto MHC class 1 is also lowest for dengue 3. Right? In contrast, the HLA class 2, which presents with CD4 T cells, is the highest for dengue 3. Right? So HLA DMA and DMB. Now, these are again A549 cells. They don't express HLA class 2 molecules that present to T cells. But DMA and DMB are those that, again, trim the invariant peptide that sits on the MHC class 2. So HLA, DMA, and B remove that peptide so that now the new antigenic peptide can be docked onto a class 2 molecule. And the HLA DOB is the inhibitor of HLA DMA and B. And so you can see that at least in A549 cells, you see an increased expression, or at least the highest expression, the least reduced expression of HLA DMA and B, whereas the DOB, dengue 3 is the lowest, right, in contrast to the others. And then finally, there's also the ligands for NK cell, activating NK cell receptors, especially NKG2D. And I thought this is interesting because there's some papers now suggesting that NK cells may be involved, and I think Ashley St. John also gave some talk along these lines. And again, with dengue 3, you're having the lowest levels, expression of MICB and the ULB-P2 and 3, all of which are ligands for NKG2D receptors. Now, you could say that, is this just differences between the serotypes? Can there be differences within dengue 3 itself? And the answer is yes. If you take the TV003 component, which is the delta 3031, and now you include it in this analysis, the delta 3031 attenuated virus actually gave rise to very high levels of interferon gamma receptor expression, which then also supported the MICB, the NKG2D ligands expression. So all that is to come to this point that, perhaps it is this difference in the serotype in the way that they cause the cells to respond and whether they express interferon gamma receptors that then affect the immunoproteasome expression that then ultimately affects what kind of T cells you develop. Right? So in 1, 2, and 4, you see that the majority of CD8 T cell epitopes are in the non-structural genes, non-structural proteins, whereas for dengue 3, it's actually in the structural proteins. Even though dengue 3 still has a lot of, in terms of sequence, share a lot of conserved regions with 1, 2, and 4, right? But it's just potentially the way in which the proteome is processed. So that's direct infection. And I want to add another level of complexity to this. I think Taya Wang earlier this morning had talked about CD16. Now here we didn't separate this into the different activating and inhibitory receptors. This is primary monocytes. The problem with ADE, of course, is that in any experiment and you do it with a virus-only infection control, then you're always struggling with imbalanced level of infection because the FC receptor-mediated pathway infection is more efficient for the virus. So what we did was then to label the virus with a fluorophore, right? And then we use flow cytometer to sort primary monocytes that show equivalent fluorophore signal, whether it's virus-only infection or ADE, and anti-mediated infection. And just to show that the signaling comes from FC receptors and not the virus, we had a heat-inactivated control. And so what you see on the left-hand side is a PCA plot, and you can see that there's a clear separation between virus-only infection and an antibody-dependent infection. And the heat inactivated control actually matches very closely with the antibody-mediated infection, suggesting that this is a lot of it is influenced by the receptors. And what are these gene expression that are changed? A lot of it has to do with splicing. So beyond the virus controlling what genes are expressed and what are not expressed, then if it comes in by the Fc receptor pathway, the splicing machinery could completely change what proteins are actually finally expressed, and therefore the function of the cell and immune response. And maybe, and I think Eva Harris also showed it earlier, talked about it earlier this morning that in her Nicaragua cohort. This is Eng Eong Ooi's data from Thailand. You can see that for primary infection, so the top 3 rows, most of it is caused by by dengue 1 and 3. There's very little dengue 2 and 4. But when you have antibodies, either in the form of maternal derived antibodies in infants or from prior infection, now you see more infection from dengue 2 especially, right? Because without it, you're now expressing all these interferon gamma receptors. Then maybe for dengue 2, it needs the Fc receptor mediated signaling to alter that response. And so with that, I'm 2 seconds over. So the question is, how will this affect Endgame? I don't know, but I think it will. But this is something that we ought to invest efforts into understanding. So with that, I'll end there, and these are the people who really contributed to this work. Thank you very much.
Thank you, Eng Eong, for a very nice presentation.